excites me most about the Pangenome Project? I'd say three things. I think firstly, in the near term it will have new ways of helping us understand human health and disease and how it relates to our genomes. So specifically, when we think about common disease, a lot of the time that is driven by genetic variants that we as a population collectively share, and the pangenome helps us find those variants and see those variants in more detail than we've been able to before. Also, for rare disease, where maybe a patient has a variant that's really, really unusual and rare and maybe causes a specific disease, we can use the pangenome to essentially sift out the things that are common and help us identify that rare variant. So it's a really good way of being able to help us essentially discriminate between the things that are common in the population and therefore unlikely to cause disease, and the things that are rare and maybe need more investigation. So that's one area. The second thing I would say is that I think the pangenome helps us see bits of the genome that we just haven't been able to sequence before. So we've got lots of new sequencing technology, and it allows us to go look at regions like the centromeres and the acrocentric chromosomes. These are portions of our genome that are very repetitive, and understand how they function. And by doing that, we're revealing new biology. We're learning about why certain bits of our genome are prone to certain kinds of translocations that can cause sterility, for example. So it's really, really illuminating and helping us kind of understand the mechanism in a really sort of fundamental way. And then the last thing I would say is that the whole purpose of the pangenome is to build this comprehensive map of genetic variation, and that is so that when we talk about variants, when we take a new sample and analyze it with respect to the pangenome, everybody is basically equally represented. We want everybody's variants, you, me, everybody, to have their variants incorporated into the pangenome structure as best we can, at least the common variants. So that the thing that we have essentially is a good representation of humanity, because that's really important for all the biomedical research that we do.
Being a part of the Human Pangenome Reference Consortium has had a big impact on my career. The effect was so deep and fast that it reminds me of the cosmic inflation after the Big Bang. I went from analyzing a fraction of a human genome from a few samples in my lab to, boom, working with many complete human genomes from many samples from many labs around the world. I owe a lot to Professor Ed Garrison, who pushed me to join the HPRC mission as a PhD student. We wanted to understand the human pangenome, but tools for that didn't exist, and so we built them. This changed my job from a software user to a software developer, to build the computational infrastructure for pangenomics, not just for us, but for everyone. During my postdoc, we explored the unknown regions of the human pangenome. We demonstrated the DNA exchange between non-homologous acrocentric chromosomes, something hypothesized for decades. And then serendipity struck. We discovered why row-persona translocations occur. These are very common in humans and are associated with infertility. This research would have been impossible without the HPRC and the way we all work together. But the HPRC changed not only my research. Having developed and applied pangenome tools, I now teach pangenomics everywhere. And my shift to education connected me with people from diverse backgrounds and cultures, and learning how to engage with different worldviews is driving me to improve as a scientist and as a person. This pangenome journey, building tools, making discoveries, teaching pangenomics, all of this catalyzed my promotion to assistant professor. Without the HPRC, I would be at a different career stage, not teaching so much, not collaborating so globally, and I would be a different person too. Friction is necessary for growth, and being a part of the HPRC is the best friction ever that shapes you into the better version of yourself.
pangenome that reflects global human genetic variation. So a lot of genomics methods require us to have a reference. The reference is something that we're comparing everyone to at the very start of our workflow. And we need this common comparison because otherwise it's just very difficult to talk to other genetic researchers about what you're doing. The problem is that it's much easier to see things that are similar to this reference than aren't similar. So if I have a picture of my face and I tell a computer to go find other faces, it'll be a lot easier to find faces that look like mine, and a lot harder to find faces that look like anyone else. And we know that we are missing a whole lot of variation. There are common variants that we are perfectly aware exist, that just aren't in our reference genome. There are things where we know the reference is basically dissimilar to nearly everyone else, but it is our reference and we're stuck with it. And our solution to this is to try to find something that represents more variation, because our single reference is an assumption. It's an assumption that one human reference is enough, and that assumption is wrong. We know that it's wrong. And so we are trying to not make assumptions. We are trying to go out into the world and sequence a bunch of people's genomes and figure out who has common variation. What common variation exists, and how can we put all that variation into one place? So we are not making assumptions about we need to have X people from Japan and X people from Africa. We are trying to just find all the common variation that exists and then collect people who have all of that into our reference pangenome. And the point of this is so that when we are sequencing anyone, if we are sequencing my genome, if we are sequencing your genome, if we are sequencing random genome from a person we found off the street, there will always be something in that reference pangenome that looks similar to whoever we're sequencing, and so we will have a good thing to compare to. We will be able to tell someone else, hey, this sample looks similar to this one in the reference pangenome with a small variation, and it's easy to find that small variation now because we know everything around it. We have something similar. And the goal of the HPRC project is just to build this common reference and release it to the world so that when all genetics researchers are trying to use a reference, they have something finally that looks similar to everyone on Earth.
My question was, how has being part of HPRC contributed to my career? And as an early career researcher, I'm a trainee, I'm a PhD student. HPRC and being part of such a big collaboration has really changed completely what I thought my PhD would be like. Very early on, in literally my second year as a PhD student, I was involved in these amazing discussions with this huge international group of scientists who are extremely supportive and all come with these different perspectives on the same science that we want to do, the same end goal that we all have. And as a student, that's really enlightening, and it broadens your interest, it broadens what you consider to be your focus in genomics. I think apart from that, it also helps that HPRC is at the cutting edge of research in pangenomics and also broadly in genomics research. And being part of HPRC means that I have access to these really good data sets. I have access to all the people who generate these data sets. I have access to using their tools. I have access to having help in generating my own tools. So in general, I think it, as an early career researcher, it has completely changed my path, and I'm really grateful to the HPRC. Thank you.
What are the new technologies that the HPRC is developing, and how are they changing, transforming genomics? So I think the main thing that we're doing is proving that you can automatically assemble genomes at scale. And automated assembly is a bit of an overstatement maybe, because there's still a lot of human intervention, quality control, these kinds of things happening. But we basically are to a place with the project where we are doing incredible data production. So the initial sequencing data is of the highest quality using the newest sequencing technologies that are available that frankly are kind of a demonstration of a total revolution in nanotechnology, where we're able to observe single DNA molecules, many single DNA molecules in parallel and at very long lengths, very high accuracy, very low error rate. And because of this, and because of innovations in sequence assembly, we're able to then derive complete assemblies of many human genomes for the first time at a truly complete level. So these assemblies span effectively from one end of each chromosome to another. And they're not perfect in this particular iteration, but they're approaching a level of perfection, of completeness, that's never been seen for hundreds of, eventually thousands of assemblies. So in result, we have this incredible foundation on which to do genomics that didn't exist before. And in response to this, because we could see it coming for a while, that these technologies would enable this, a lot of members of the HPRC have been developing information technologies that work downstream of these. They make it possible to relate these sequences to each other, we call that alignment, to build representations of the many-way alignment between these genomes. We tend to call these graphs, pangenome graphs. And then we reuse these to do analyses, to understand evolution, of parts of the genome that weren't known before. So I guess that gets to the second part of the question, is how is this transforming genomics? And at the moment, it's subtle. So if you go to big genomics conferences, you do see use of these resources, but it's sort of the tip of the iceberg, I think. We're just beginning to see uptake in the biomedical community, in human medicine, and also in biology in general. In some species, people really need a pangenome in order to operate because there's high diversity, because the genomes they're working with are very different than each other. In humans, we've been able to get by on this kind of crutch that everything is kind of like the reference, and we just have to have one reference that represents the whole genome, and we can align short reads against it and understand lots about biology. But what we've shown in this project is that that is not the case. There are parts of the genome that were invisible. We've made them visible, and using that, we've solved basic questions in chromosome biology, for example. Recent work that I've been involved with demonstrated the reason that 1 in 800 people is born with a translocation between two chromosomes that connects them into a fusion. It's called a Robertsonian chromosome. This can impact fertility, and it's really big. So we've seen it for 50 years or more on cytogenetic microscopes. And only now we understand why it's happening, because there are sequences that are homologous, they're similar to each other on these chromosomes that recombine in this way, that foster this recombination. We saw that in the Human Pangenome data for the first time. And because of that, we're able to actually confirm that this was the case. So this is one small but textbook-altering example of the way that our work is having an impact in genomics.
So how will HPRC research advance our understanding of human health and disease? So clinical genetics is quite complex. When we see patients in the clinic for presumed genetic diagnoses, we often send off genetic testing to figure out not only can we identify the genetic basis of their disease, but also find a potential therapeutic avenue. We know that when we do this, that there's a lot that we can look at, and there's a lot that we just don't understand about the human genome. And part of that is because there's a lot of really complex areas of the human genome which matter a lot in human health and disease, but we just don't have the tools to appropriately investigate that. One of the things that excites me the most about the HPRC is it really is giving us that toolkit for now starting to look at some of these areas of the genome which we know are medically important and play a huge role in disease, but for which we've previously just been unable to appropriately assess. And I think that this will really have a big, profound impact in terms of how we get genetic diagnoses for the individuals we're seeing in clinic, and not only that, but how we move those genetic diagnoses into improved therapeutics to help improve their outcomes.
The question is, how is HPRC continuing the legacy of the Human Genome Project? With your permission, I'd like to kind of change the question to sequencing and computation and the emergency idea of the Human Genome Project and where we are today. And what I was going to observe is DNA sequencing was invented while I was in medical school, and the personal computer came out about 5 years later, and the Macintosh came in 4 years after that. So this combination of computers and DNA sequencing technology all converged in that period. And then that, of course, gave rise to this idea of having a Human Genome Project, the goal of which was to map and sequence the human genome as a tool for understanding human biology, but also to improve clinical care by being able to derive information that you can't get any other way, and that DNA is the storage and transmission medium for genetic information, and it's therefore kind of a bedrock of human biology. At the time, it was kind of a crazy idea to do the Human Genome Project. The technology was not anywhere close to being ready for doing something the size of the human genome. It was barely able to do even small viral genomes. But it turned out that the technology did move fast enough that it became a practical reality to do the Human Genome Project. So from 1985 to 1990, the project idea was kind of gestating, and then of course officially it began in 1990. And the idea there was to do a reference map and sequence. And the result of that was this composite sequence that we've been using since, really since 2000, well, actually even before the first original completion of the human genome in 2003. We've been using that linear reference genome that's a composite of many people, 70% of it coming from one guy in Buffalo. And what HPRC is doing now is creating a new reference resource that is both more complete, more accurate, and is moving away from this fantasy that there is a reference human genome, because there are going to be a lot of reference genomes. And the idea is to accommodate the diversity and the variation that exists among human populations. And HPRC is a huge step in that direction. It's the US element, and I think it will evolve, I hope, into a truly global public good, a resource that everybody can use everywhere for advancing science, but also it's going to be, I think, the bedrock of clinical sequencing. And clinical sequencing it's already really important for cancer and rare disease, but it's going to be spreading to all of medicine, not as the cure-all or anything like that, but as a major source of information that's really, really practically useful for clinical care. And I think so, HPRC I think is kind of setting the stage for the next couple decades of technological evolution and the incorporation of DNA sequence information into clinical decision making.
A little bit of background. My dissertation work all dealt with finding allele-specific expression as a way to find cisgenetic effects. But at the time I worked in mouse models, so it was 2 haplotypes. It's much easier to do things, and I really wanted to move into humans, and I really wanted to move into a more realistic, more complex system. And what that meant, even at the time, this was before I got involved with the HPRC, it meant that I needed to go from a linear-based method to some sort of more sophisticated methodology, like a graph-based method. And it was at that time that I was sort of, given the opportunity to do a postdoc with Ting and become a member of the HPRC. And so I did it, took the leap. And I would say that my participation in the HPRC and collaborations with the people have really fundamentally shaped how my research has developed in a couple of ways, right? When you're going from mouse complex trait genetics and linear space to like human genetics and graph space, that's a big methodological shift. And so simply having peers that I could reach out that I knew were experts in the tools or had developed the tools has been, incredibly empowering, because I could just message, Glenn or Adam or, Christian as to a specific thing and get feedback from people. And that has, really guided not only the methods that I've pursued to develop myself and how I've sought to apply the pangenome, but I think more fundamentally, having these people to talk to, sitting in on the seminars, listening to all of this work has really fundamentally changed the sorts of questions that I even consider asking, right? Like it's rewired how I think about genomes, as a whole, and natural variation, and the methodology that needs to go along with answering questions of the consequences of natural variation, say in humans.